There is a moment in the shallows of a river in sub-Saharan Africa, or in the tidal estuaries of northern Australia, or along the muddy banks of the Ganges Delta, when the water does something that water is not supposed to do. It moves with intention, a ripple that has no wind behind it, a displacement of surface that originates not from current or tide, but from something below, something that has been lying perfectly still for so long that the water itself had forgotten it was there. And then, in a fraction of a second that the human eye can barely resolve, the stillness becomes an explosion. Jaws that can generate more force than any other living animal on Earth break the surface. Something that was alive a moment ago is no longer, and the water closes over the event as though nothing happened at all. This is the crocodile, and it has been doing exactly this in more or less exactly this way for longer than the mind can comfortably hold. Not for a few thousand years, not for a few million years, but for 200 million years, a span of time so vast that it encompasses the rise and fall of the dinosaurs, the breakup of the supercontinent Pangaea, multiple mass extinction events that reset the biological history of the planet, the drift of continents to their present positions, the elevation of mountain ranges that did not exist when the first crocodilians appeared, and the entire evolutionary history of every mammal, bird, and flowering plant that currently inhabits the earth. Through all of it, the crocodile endured. Through all of it, the crocodile barely changed. That fact, the fact of the crocodile's extraordinary temporal persistence, its 200 million year tenure on a planet that has shown no hesitation in eliminating lineages far more numerous and seemingly more successful, is the central mystery of this animal and the starting point for everything that follows. Why? Why has an animal that looks, to the casual eye, like a leftover from a more primitive era of life on Earth, outlasted everything that the history of the planet has thrown at it, including the catastrophic asteroid impact 66 million years ago that eliminated 3/4 of all species alive at the time, including every non-avian dinosaur? Why has an animal that has changed so little, persisted while so much around it, changed so dramatically? And what does the answer to that question tell us about the nature of biological success, about the relationship between simplicity and resilience, and about what it means to be perfectly adapted to a world that never stops changing? The answers are not simple, and they are not what most people expect. The crocodile is not a primitive animal in any meaningful biological sense. It is not a leftover, not a relic, not a survivor by accident or by luck. It is, in the precise vocabulary of evolutionary biology, an organism of extraordinary sophistication, one whose apparent simplicity conceals layers of physiological, behavioral, and ecological complexity that researchers are still actively uncovering. To understand the crocodile is to revise almost every assumption that its ancient, armored, cold-blooded appearance invites. It is to encounter an animal that is not what it looks like, not what it seems, and not what 200 million years of popular mythology have made it out to be. Let us begin at the beginning, or as close to the beginning as the fossil record allows. The archosaurs, the group of reptiles from which both crocodilians and dinosaurs descend, first appeared in the early Triassic period, approximately 250 million years ago. In the aftermath of the most severe mass extinction in Earth's history, the Permian-Triassic extinction event, which eliminated an estimated 90 to 96% of all marine species and 70% of all terrestrial vertebrate species. The world that the archosaurs colonized was, in a real sense, an empty world, a planet whose ecological communities had been devastated and whose niches were wide open, available to whichever lineages could move into them most effectively. The archosaurs diversified rapidly in this open landscape. By the Middle Triassic, the group had split into two major lineages: the bird-line archosaurs, which would eventually give rise to the dinosaurs and, through them, to modern birds, and the crocodile-line archosaurs, which would give rise to the crocodilians. The early crocodile-line archosaurs were not aquatic. This is one of the most persistently surprising facts about crocodilian evolutionary history, that the ancestors of an animal so perfectly suited to a semi-aquatic life began their evolutionary journey as terrestrial animals. Some of them bipedal, some of them armored in ways that their descendants have partially retained, ranging across the dry landscapes of the Triassic in forms that would be largely unrecognizable to someone familiar only with living crocodilians. The transition to the semi-aquatic lifestyle that defines modern crocodilians was gradual, unfolding over tens of millions of years through a succession of intermediate forms that the fossil record has preserved in enough detail to trace the broad outlines of the story. By the late Triassic and early Jurassic, approximately 200 million years ago, crocodile-line archosaurs were diversifying into a range of ecological roles, including fully marine forms that swam in the shallow seas of the Jurassic and Cretaceous with tail flukes and paddle-like limbs, terrestrial forms with limbs positioned directly beneath the body like those of mammals, and semi-aquatic forms that increasingly resembled the modern crocodilian body plan. It is from these semi-aquatic forms that the living crocodilians descend, and it is in them that the suite of anatomical characteristics that define the group, the long, flattened snout, the laterally compressed tail, the elevated eyes and nostrils, the osteoderms of the back, the sprawling limb posture, began to take shape. The modern crocodilian order, Crocodilia, containing the living species we know today, first appeared in the fossil record in the late Cretaceous, approximately 80 million years ago. These animals survived the end Cretaceous mass extinction, the event triggered by the asteroid impact at Chicxulub 66 million years ago, when the non-avian dinosaurs, the marine reptiles, the pterosaurs, and enormous numbers of other lineages did not. The reasons for their survival while so many other lineages perished are not fully understood, but current research points to several factors. Their semi-aquatic lifestyle may have provided access to food resources, carrion, fish, invertebrates, that remained available through the ecological disruption that followed the impact. Their ability to enter torpor during periods of food scarcity may have allowed them to survive the prolonged darkness and cold that resulted from the impact's dust cloud, and their relatively low metabolic requirements compared to similarly sized warm-blooded animals may have made them less vulnerable to the food resource collapse that eliminated so many other lineages. Whatever the reasons, the crocodilians emerged from the end Cretaceous extinction as survivors, and they have been diversifying, adapting, and persisting ever since through the Paleocene, Eocene, Oligocene, Miocene, Pliocene, and Pleistocene into the present, a continuous lineage spanning 66 million years of post-extinction history, added to the approximately 130 million years of crocodilian-like evolution that preceded it, 200 million years, give or take, of crocodiles being crocodiles. The living crocodilians comprise 25 species, divided among 3 families: the true crocodiles, family Crocodilidae, with 15 species; the alligators and caimans, family Alligatoridae, with 15 species, the alligators and caimans. Family Alligatoridae, with 8 species, and the gharials, family Gavialidae, with 2 species including the critically endangered Ganges gharial and the false gharial of Southeast Asia. These 25 species are distributed across the tropical and subtropical regions of every inhabited continent, except Europe, occupying freshwater rivers, lakes, swamps, estuaries, coastal mangroves, and in the case of the saltwater crocodile, the open ocean for significant distances. The saltwater crocodile, Crocodilis porosus, deserves early and particular attention because it is, by multiple measures, the most extraordinary of the living crocodilians, and in several respects, the most extraordinary reptile alive on Earth. It is the largest living reptile, with mature males regularly exceeding 5 meters in length, and occasionally reaching 6 meters or beyond, and weighing over 1,000 kilograms. It is the most widely distributed crocodilian, ranging from the eastern coast of India through Southeast Asia and the Indonesian archipelago to northern Australia and the western Pacific Islands. A distribution maintained not by land travel, but by open ocean swimming, which the saltwater crocodile undertakes with a facility that still surprises researchers who track their movements by satellite. Individuals have been recorded crossing hundreds of kilometers of open ocean, navigating between island groups, and appearing in locations far from their known home ranges in ways that imply a degree of spatial awareness and navigational ability that raises profound questions about the cognitive capacities of an animal, not traditionally regarded as particularly intelligent. The saltwater crocodile is also unambiguously the most dangerous animal to human beings of any living crocodilian, and in regions where it coexists with human populations without adequate protective infrastructure, one of the most dangerous animals on Earth, period. It is not aggressive in the way that some animals are aggressive, not prone to charging or threatening behavior as a first response. It is something more calculated than aggressive, an ambush predator of extraordinary patience and precision that treats large, warm-blooded mammals, including humans, as prey items, and that has the physical capacity to kill and consume an adult human being with an efficiency that is simultaneously terrifying and biologically logical. Hundreds of attacks on humans are recorded each year across its range, with fatality rates that vary by region but that are consistently significant. Understanding why these attacks occur, what drives them, and how the risks can be reduced without eliminating the crocodile is one of the most practically important questions in crocodilian biology, and it requires understanding the animal's sensory systems, behavioral ecology, and cognitive processes at a level of detail that research has only recently begun to provide. Before examining the modern crocodilian in the detail its biology demands, it is worth spending time with the physical reality of the animal, its body, its structure, its mechanical systems, because these are the foundation on which everything else rests, and because they are considerably more sophisticated than their ancient reptilian appearance suggests. The skin of a crocodilian is not simply armor, though it is extremely effective armor. It is a complex, multifunctional organ system. The osteoderms, the bony plates embedded in the skin of the back and sides, provide protection against the teeth and claws of conspecifics and other predators, and they serve as thermal mass, absorbing heat from the environment and releasing it slowly, contributing to the animal's thermoregulatory efficiency. But the skin also contains integumentary sensory organs, small, dome-shaped structures distributed across the head and body, particularly concentrated around the jaws. that are among the most sensitive mechanoreceptors known in any vertebrate. These organs, sometimes called dome pressure receptors or DPRs, can detect minute disturbances in water. The pressure waves generated by a fish moving at a distance of several meters. The surface ripple created by a small mammal stepping to the water's edge, the vibration transmitted through water by the footfall of a large animal approaching the bank. They allow a submerged crocodilian to perceive its aquatic environment with a sensitivity that supplements, and in some conditions exceeds, the information available from its eyes and ears. The jaws deserve extended examination because they are the tool around which the crocodilian's predatory strategy is organized, and because they are more precisely engineered than popular representations typically convey. The skull of a crocodilian is a mechanical marvel, a structure evolved over hundreds of millions of years to deliver enormous bite force while remaining light enough not to impair the animal's ability to remain neutrally buoyant in water. The temporal muscles that drive jaw closure are massive, filling the entire posterior portion of the skull, and they generate forces that have been measured in large individuals of Crocodilus porosus at more than 3,000 pounds per square inch, the highest bite force recorded for any living animal. For comparison, a large lion generates approximately 600 pounds per square inch. A hyena, which is specifically celebrated for its bone-crushing bite, generates approximately 1,100. The saltwater crocodile generates forces nearly 3 times greater. This force is not evenly distributed along the jaw. The teeth of crocodilians are not cutting tools. They are not designed to slice or sever like the carnassial teeth of carnivorous mammals. They are conical, robust, interlocking pegs designed to grip and hold. The strategy is not to cut prey apart, but to seize it with a grip that cannot be broken, and then to rotate. The famous and terrible death roll, spinning the body along its longitudinal axis with enough force to disjoint or dismember the prey item, or to drag it underwater, where drowning accomplishes what teeth and force alone cannot. The death roll is not a behavioral oddity. It is a precisely functional killing technique optimized for prey that is too large to be subdued by grip alone, and it is executed with a mechanical efficiency that reflects the millions of years of selection pressure that produced it. The asymmetry between the power of jaw closing and jaw opening is one of the most remarked upon facts about crocodilian anatomy. The muscles that close the jaw are extraordinarily powerful. The muscles that open it are relatively weak. This means that a human being of average strength can hold a crocodile's mouth closed with their bare hands. A fact that is true and that is regularly demonstrated in educational and research contexts. What this asymmetry does not mean is that the animal is therefore easily managed or controlled, because the rest of its body, the tail, the limbs, the twisting torso, is powerful to a degree that no human grip can resist. The jaw asymmetry is simply a reflection of the animal's predatory strategy. Maximum power in the direction of capture, minimum metabolic investment in the direction that matters less. The teeth themselves are regularly replaced. Throughout the crocodilian's life, a system called polyphyodonty, in which each tooth position contains a functional tooth, a replacement tooth developing beneath it, and a further replacement developing beneath that. Crocodilians may replace each tooth position 50 or more times over a lifetime. This ensures that the gripping tools on which their predatory success depends remain sharp and structurally sound regardless of the wear and occasional breakage that a lifetime of hard use inevitably produces. The biological engineering of this system, the precise hormonal and developmental signaling that coordinates replacement tooth development with functional tooth wear, is a subject of active research, partly because understanding it has potential implications for regenerative medicine in humans whose tooth replacement system produces only a single set of adult teeth. The digestive system of the crocodilian is adapted to process prey items with a thoroughness that is remarkable even by the standards of efficient carnivores. The stomach is highly acidic. pH values in the range of 1 to 2 have been recorded in crocodilian stomach contents comparable to battery acid, enabling the digestion of bone, hair, shell, and other materials that the digestive systems of most vertebrates cannot process. Crocodilians routinely swallow stones, gastroliths, which were once thought to function as ballast for buoyancy control but are now understood to serve primarily as grinding surfaces in the stomach, mechanically assisting the breakdown of hard food items in combination with the chemical action of the gastric acid. The efficiency of crocodilian digestion means that almost nothing of a consumed prey item is wasted. Shells are broken down. Even the keratinous elements of prey, hooves, horns, claws, are processed over time. The metabolic physiology of crocodilians is one of the most distinctive and ecologically significant aspects of their biology, and it is one of the areas where the popular conception of the animal as simply a cold-blooded, slow metabolizing reptile falls most dramatically short of the reality. Crocodilians are ectotherms. Their body temperature is determined primarily by the external environment rather than by internal metabolic heat production, as in mammals and birds. This is true, but the popular extrapolation from this fact, that they are therefore metabolically simple, physiologically primitive, and limited in their biological capacities, is substantially wrong. The metabolic rate of a resting crocodilian is indeed low by mammalian standards, perhaps 5 to 10% of what a similar-sized mammal would require. But this is not a constraint. It is an adaptation, a profoundly useful biological property that allows the animal to survive extended periods without food that would be fatal to a mammal of equivalent size. A large crocodile can go without eating for a year or more and survive, drawing on fat reserves stored in the tail and elsewhere in the body. This fasting tolerance is not simply dormancy. The animal remains physiologically active during fasting. Its immune system functions, its wound healing continues, its sensory systems remain operational. It is simply operating at an extremely low metabolic rate, spending energy at a rate that its reserves can sustain indefinitely. When food is available, however, the crocodilian's metabolic system can upregulate dramatically. An ability to shift from extremely low to much higher metabolic activity that has been described by researchers as one of the most impressive metabolic flexibilities documented in any vertebrate. After a large meal, a crocodilian's digestive system ramps up to a state of intense activity, processing the meal rapidly and storing the energy with extraordinary efficiency. The heart, which in crocodilians is four-chambered uniquely among non-avian reptiles, allowing complete separation of oxygenated and deoxygenated blood, can adjust its output and the routing of blood flow through the body in response to metabolic demand in ways that are more sophisticated than those of most reptiles. And that approach, in functional terms, the cardiovascular flexibility of mammals and birds. The four-chambered heart of crocodilians is worth dwelling on because it is one of the most revealing facts about the evolutionary history of the group. A four-chambered heart in which oxygenated blood from the lungs and deoxygenated blood from the body are completely separated is a characteristic otherwise found only in mammals and birds. The two groups of vertebrates that evolved endothermy, the ability to generate their own body heat internally. The presence of a four-chambered heart in crocodilians tells us that this architecture evolved before the divergence of the Archosaur lineages, and that the common ancestor of crocodilians, dinosaurs, and birds already possessed it. The birds retained it as part of the physiological suite that supports their high-energy, warm-blooded lifestyle. The crocodilians retained it, but deployed it in service of a very different physiological strategy, one optimized not for sustained high metabolic output, but for extreme metabolic flexibility, the ability to shift between states of very low and relatively high activity as circumstances demand. This metabolic flexibility is the key to understanding how crocodilians have survived everything the history of the planet has thrown at them. Animals with high fixed metabolic rates, mammals, birds, are vulnerable to food resource collapses because they cannot reduce their energy expenditure below a certain baseline. When food becomes scarce, they starve. Crocodilians, with their ability to reduce metabolic activity to a fraction of baseline, can survive through resource collapses that eliminate their warm-blooded competitors. This is almost certainly part of the explanation for their survival through the end Cretaceous extinction, when the global food web was severely disrupted for months to years following the asteroid impact. The dinosaurs, with their high metabolic demands, could not survive the food shortage. The crocodilians, their metabolic engines throttled down to near zero, could wait it out. The sensory systems of crocodilians are another area where careful research has revealed capabilities far more sophisticated than earlier, simpler models suggested. The eyes are positioned on the top of the skull, allowing the animal to maintain visual surveillance of its surroundings while the rest of the body is submerged. A classic semi-aquatic adaptation found in many animals that straddle the boundary between aquatic and terrestrial environments. But the eyes themselves are more capable than this simple description implies. They are equipped with a tapetum lucidum, the reflective layer behind the retina that produces eye shine in photographs and that amplifies light sensitivity in low light conditions. That makes crocodilians highly effective nocturnal hunters. The slit-shaped pupils, which can dilate to large circles in darkness, allow the eyes to gather maximum light under conditions where human vision would be essentially useless, and the ability to see well both in air and underwater, accommodated by adjustments in the shape of the lens, gives the crocodilian a visual range across the air-water boundary that few other predators can match. The hearing of crocodilians is acute for a reptile, and it plays a particularly important role in their social and reproductive behavior. They are among the most vocal of reptiles, producing a range of sounds from the deep subsonic rumbles used in territorial and reproductive communication, felt as vibrations through the water as much as heard through the air, to the higher-pitched distress calls of juveniles, which reliably trigger protective behavior in adult females. The ability to detect and respond to sounds both in air and transmitted through water is an adaptation that serves both predatory and social functions, and the neural processing that underlies it is more sophisticated than the simple reptilian label would suggest. The olfactory system is powerful and forms an important component of the crocodilian sensory picture of its environment, particularly for detecting prey and conspecifics at range. Chemical signals in water, blood, metabolic byproducts, scent compounds released by potential prey are detected through both the olfactory organs in the nasal passages and through the Jacobson's organ, a chemosensory structure in the roof of the mouth that processes molecules brought into the mouth by the tongue. The integration of information from these multiple sensory channels, visual, tactile, auditory, olfactory, and the pressure-sensitive integumentary sensory organs, gives the crocodilian a comprehensive, multidimensional picture of its environment that enables the precision of its predatory strikes even in conditions of very low visibility. Understanding all of this, the evolutionary history, the physical structure, the physiological sophistication, the sensory capabilities, is the foundation on which an understanding of crocodilian behavior must be built. The behavior of these animals is not simple. It is not the mechanical stimulus-response behavior that older models of reptilian cognition implied. It is flexible, context-dependent, individually variable, shaped by learning and experience, as well as by instinct, and in some dimensions more cognitively complex than researchers were prepared to find in an animal whose evolutionary lineage diverged from that of mammals more than 300 million years ago. The predatory behavior of crocodilians is the dimension most familiar to popular audiences, and it is worth examining in detail not because it is the most important aspect of their biology, but because it is the lens through which most people encounter the animal, and because the reality of how crocodilians hunt is considerably more interesting than the simple image of an ambush predator waiting in the shallows for an unwary prey animal to approach. Crocodilian predation is opportunistic in the broad sense. These animals will take whatever prey is available, appropriately sized and accessible. But it is far from random or unsophisticated at the level of individual hunting events. Long-term observation of Nile crocodiles at river crossings in the Serengeti Mara ecosystem of East Africa, where the annual wildebeest migration brings hundreds of thousands of animals to a small number of crossing points, has revealed behaviors that go well beyond simple ambush. Crocodiles position themselves at crossing points in ways that reflect knowledge of current patterns, water depth, and prey approach routes. Positioning that is not random, but that maximizes the probability of successful contact. Individual crocodiles have been documented returning to the same positions at the same crossing points year after year, suggesting spatial memory and site fidelity that implies learning from past experience. Groups of crocodiles at crossing points engage in what appears to be coordinated behavior, not true cooperation in the mammalian sense, but a spatial organization that collectively increases hunting efficiency beyond what individual animals could achieve alone. The question of whether crocodilians are capable of true cooperative hunting, the intentional coordination of individual behaviors toward a shared predatory goal, is one of the most actively debated in crocodilian behavioral science. Documented cases of multiple crocodiles simultaneously attacking large prey, or of individuals apparently herding fish toward each other, have been interpreted by some researchers as evidence of genuine cooperation. Others argue that these observations can be explained by simultaneous individual responses to the same prey item without any need to invoke intentional coordination. The debate reflects genuine uncertainty in the data and genuine disagreement about the cognitive requirements of different behavioral interpretations, a disagreement that is unlikely to be resolved quickly. Because observing crocodilian behavior in sufficient detail to distinguish true cooperation from simultaneous individual response requires a level of field observation that is technically demanding and logistically challenging. What is not in dispute is that crocodilians learn from experience in ways that adjust their behavior, that individual animals over a lifetime accumulate knowledge about the specific features of their environment, the behavior of prey species, and the consequences of different hunting approaches, and that this accumulated knowledge influences their hunting behavior in ways that make experienced individuals meaningfully more effective hunters than naive ones. This learning capacity is one of the lines of evidence for a cognitive sophistication in crocodilians that challenges the older, simpler model of reptilian intelligence, and it connects to a broader reconsideration of reptilian cognition that has been gaining momentum in behavioral science for the past 2 decades. The question of crocodilian intelligence is one that science has approached slowly and with considerable caution, partly because intelligence is a concept that is notoriously difficult to define, even in species closely related to ourselves, and partly because the history of comparative psychology is littered with examples of researchers either overestimating or underestimating the cognitive capacities of animals based on assumptions derived from their own evolutionary heritage, rather than from the specific demands of the animal's ecological niche. The tendency to assume that large brain size is a prerequisite for complex behavior, for instance, has been repeatedly challenged by demonstrations of sophisticated cognition in animals with small brains. Bees, fish, cephalopods, whose neural architectures solve complex problems through mechanisms very different from those of the mammalian cortex. Crocodilians have relatively small brains by mammalian standards, and the regions of the brain associated with complex cognition in mammals, the neocortex and its analogs, are less developed in reptiles. For most of the 20th century, this neuroanatomical observation was taken to imply that crocodilian behavior was largely stereotyped and inflexible, driven by innate programs with little capacity for learning or adjustment. This view is now understood to be significantly wrong, not because the neuroanatomical observations were incorrect, but because the inference from brain structure to behavioral capacity was unjustified. Different neural architectures can support functionally similar behavioral outcomes through different computational mechanisms, and the relevant question is not whether a crocodile's brain looks like a mammal's brain, but whether the crocodile's behavior shows the hallmarks of flexible, experience-dependent processing that we associate with intelligence. By this behavioral criterion, the evidence for meaningful cognitive complexity in crocodilians has been accumulating steadily. Problem-solving experiments conducted in controlled settings have shown that crocodilians can learn to associate specific stimuli with specific outcomes and can generalize this learning to novel stimuli. Individual recognition, the ability to distinguish specific other individuals from the same species, has been documented, which requires a memory system capable of storing and retrieving specific, complex perceptual representations. Play behavior, which is in most vertebrates associated with cognitive flexibility and the development of behavioral repertoires, has been documented in crocodilians in contexts that appear to go beyond functional explanation. Adults interacting with objects or engaging in locomotor play in ways that have no obvious, immediate survival benefit. The documentation of play in crocodilians is particularly interesting because it adds these animals to a list of vertebrate groups where play behavior has been confirmed that is broader than was recognized even 15 years ago, and because it suggests that the motivational and neural systems that support play in mammals and birds, systems associated with curiosity, exploration, and the intrinsic reward of novel interaction with the environment, have deeper evolutionary roots than previously thought. If crocodilians play, and if crocodilians share a common ancestor with mammals through a lineage extending back to the earliest amniotes, then the capacity for play may be an ancient vertebrate characteristic whose expression has been shaped differently by different evolutionary trajectories, rather than a derived characteristic of warm-blooded, animals that evolved independently in mammals and birds? The social behavior of crocodilians is more complex and more structured than popular representations typically suggest, and it operates through a communication system that spans multiple sensory channels and that encodes a richer variety of information than earlier researchers recognized. Crocodilians are not as social animals that simply compete for resources and mates without meaningful social interaction. They have social hierarchies maintained through a combination of size-based dominance and experience-based learning. That structure access to resources, feeding, sites, basking positions, nesting areas. In ways that reduce costly direct conflict, they have communication systems involving visual displays, vocalizations, vibrational signaling through the water surface, and chemical signals that convey information about identity, size, reproductive status, and motivational state. And they have parental behaviors, particularly in females, but also in some species in males, that represent some of the most sophisticated parental care documented in any reptile. The maternal behavior of crocodilian females is particularly remarkable and deserves careful description because it is so far removed from the popular image of reptiles as animals that lay eggs and abandon them, leaving their offspring to fend for themselves. Female crocodilians construct nests, either mound nests of vegetation and organic material in the case of most crocodile species, or whole nests excavated in sandy banks in the case of some species, and then guard those nests continuously for the entire incubation period, which lasts approximately 70 to 90 days, depending on species and environmental conditions. During this period, the female remains close to the nest, defending it against potential predators, monitor lizards, mongooses, large birds, other crocodilians, and in some areas, humans, with a ferocity that represents a significant reversal of the animal's usual avoidance behavior around large threats. At the moment of hatching, the female responds to the high-pitched chirping calls of the emerging young by excavating the nest if necessary, gently picking up the hatchlings in her jaws. Jaws capable of exerting over 3,000 pounds per square inch of force, now being used with enough delicacy to carry a 10-centimeter hatchling without injury and carrying them to the water. This behavior, requiring the precise motor control to modulate jaw pressure from maximum force to near zero based on the nature of the object being held, is a striking demonstration of the behavioral flexibility that underlies the apparent simplicity of crocodilian anatomy. The female continues to guard the young for weeks or months after hatching, and the hatchlings produce distress calls that reliably elicit protective responses from the female. In some species, males also participate in nest guarding and hatchling transport, a degree of biparental care that is unusual among reptiles and that speaks to the social complexity of these animals. The hatchlings themselves are not helpless. From the moment they enter the water, they are miniature versions of the adult body plan, capable of swimming, hunting small prey, and responding appropriately to threat. But they are extremely vulnerable to predation by birds, fish, larger crocodilians, including adults of their own species, monitor lizards, and numerous other predators that find the small, protein-rich hatchlings attractive prey. Mortality in the first year of life is extremely high. Estimates suggest that fewer than 10% of hatchlings survive to their first birthday in most populations. The survivors grow rapidly at rates determined by food availability and temperature, and juvenile crocodilians that successfully navigate the first few years of life. When their small size makes them vulnerable to a wide range of predators, enter a phase of dramatically reduced mortality risk as their size increasingly exceeds the capacity of most predators to subdue them. The growth trajectory of crocodilians is one of their most ecologically significant characteristics because it means that individuals that survive long enough grow into a size class that has essentially no natural predators. An adult male saltwater crocodile of 5 meters and 800 kilograms has no natural enemy in any ecosystem it inhabits. It is the apex predator without qualification, and it occupies that position not through speed or agility or social cooperation, but through the combination of size, armored body, immense bite force, and predatory precision that makes it the most dangerous large animal in any river system, estuary, or coastal area it inhabits. This apex status, rare among living animals in its completeness, is not incidental to the crocodilian's ecological role. It is the foundation of that role, and understanding it is essential for understanding the consequences of crocodilian population changes for the ecosystems they inhabit. The ecology of crocodilians as apex predators generates effects that cascade to entire ecosystem food webs in ways that are beginning to be understood in detail. As keystone predators, crocodilians regulate prey populations in ways that prevent competitive exclusion and maintain diversity. Their physical activities, nest building, burrowing, pool excavation, modify habitats in ways that create resources for other species. Their kills, the portions of carcasses they do not consume, and the carcasses of animals they kill but do not retrieve, provide food for a community of scavengers. And their role as both predators and prey at different life stages means that they participate in food web dynamics at multiple trophic levels simultaneously. In the river systems of sub-Saharan Africa, the Nile crocodile is the dominant aquatic predator, and its presence structures the behavior of virtually every other large animal that uses those rivers. Hippopotami, which are larger than crocodiles and which coexist with them without apparent fear, are exceptional in their indifference to crocodilian presence. For most other animals, the riverbank is a place of acute risk where drinking and crossing must be accomplished quickly and alertly. This behavioral wireness, the landscape of fear imposed by a predator on the behavior of potential prey, has ecological consequences that extend beyond the direct predation events themselves. It determines where and when different prey species use water resources, which in turn influences the spatial distribution of grazing pressure on bankside vegetation, the degree of erosion at different points along the river, and the availability of habitat features used by smaller species. Remove the crocodile, and the behavioral ecology of the entire river system changes. This has been documented in areas where crocodile populations were severely reduced by commercial hunting during the mid-20th century. The large-scale commercial hunting of crocodilians for their skins, the smooth, beautifully patterned belly skin of saltwater crocodiles and caimans, and the similarly valued skin of the Nile crocodile reached its peak in the 1950s and 1960s, when the fashion industry's demand for exotic leather drove harvests of millions of animals annually across the species ranges. The consequences for crocodilian populations were severe. Several species, including the American crocodile, the Orinoco crocodile, and the gharial, were reduced to fractions of their historical abundances. The saltwater crocodile was essentially eliminated from large portions of its Australian range. The Nile crocodile was dramatically reduced in many African river systems. Where these reductions occurred, ecological changes followed. Fish populations in some river systems increased dramatically in the short term, a response to reduced predation pressure, but then experienced boom and bust population dynamics as the regulatory function of the apex predator was removed and competitive interactions among fish species intensified without the top-down control that the crocodile had provided. Bankside vegetation patterns changed as the behavioral landscape of fear was modified. Water quality in some systems declined as the scavenging function of crocodilians. Their removal of carcasses from river systems was reduced. These observations from multiple river systems across multiple continents provided some of the earliest empirical evidence for the ecological importance of apex predators in freshwater systems, and they contributed to the broader scientific recognition of trophic cascades that has since been documented in terrestrial, marine, and freshwater ecosystems around the world. The recovery of crocodilian populations following the imposition of legal protections in the 1970s and 1980s, one of the most significant conservation successes in 20th century wildlife management, provided a natural experiment in reverse, documenting what happens when apex predators are restored to systems from which they had been largely removed. In northern Australia, where the saltwater crocodile was protected by law in 1971, the species' recovery from near elimination to current population sizes estimated at 100,000 individuals or more has been dramatic and has occurred more quickly than most conservationists predicted. The recovery has been accompanied by observable changes in fish community dynamics, bankside vegetation, and the behavior of other large vertebrates in the rivers and estuaries where crocodiles have returned. The Australian saltwater crocodile recovery story is remarkable for several reasons beyond the simple fact of population recovery. It demonstrates that a large, long-lived apex predator can recover to ecologically functional abundance within a few decades if the direct causes of population decline are effectively addressed and if adequate habitat and prey resources remain. It demonstrates that communities can adjust to coexistence with a large and genuinely dangerous predator, though this adjustment is not without cost and conflict. And it demonstrates that the legal and regulatory frameworks developed under the international conservation agreements of the 1970s, particularly CITES, the Convention on International Trade in Endangered Species, can be effective in reducing commercial hunting pressure when adequately enforced. The Australian experience also illustrates the genuine complexity of human-crocodile coexistence in regions where the recovered predator and human communities share the same rivers, estuaries, and coastal areas. Crocodile attacks on humans in the Northern Territory, Queensland, and Western Australia have increased as the population has recovered. Not because the crocodiles have become more aggressive, but simply because there are more of them, they are larger, and the overlap between their habitat use and that of humans, swimming, fishing, camping along watercourses, provides more frequent opportunities for encounters. The management of this coexistence, the balancing the ecological and conservation value of the recovered population against the safety needs of the human communities that live alongside it, is an ongoing challenge that involves scientific monitoring, public education, physical infrastructure at high-risk locations, and in some cases, the removal of individual crocodiles that have established territories in areas of high human use. The question of how human communities learn to coexist with large, potentially dangerous wildlife is one of the most important and most difficult in conservation, and the crocodile presents this question in an unusually acute form because it is not simply a predator that might occasionally cause harm, but an animal that in appropriate conditions actively preys on humans, This is not a reframable behavior. It is not a misidentification, a territorial response, or a defensive attack. Categories that apply to most large animal attacks on humans and that suggest management approaches focused on reducing the trigger conditions. Crocodilian predation on humans is genuine predation in which the human being is recognized as suitable prey and targeted accordingly. Understanding and accepting this fact honestly, without either dismissing the risk or exaggerating it to the point of demonizing the animal, is a prerequisite for effective management of human-crocodile coexistence. The distribution of crocodile attack risk is not uniform across the species range, and understanding the factors that determine risk levels in different areas is essential for directing management effort efficiently. Attack rates are highest in areas where large crocodilian populations overlap with human communities that use waterways extensively for subsistence activities, fishing, bathing, collecting water, washing clothes, without adequate protective infrastructure such as raised jetties, wire enclosures, or cleared sightlines at water access points. They are lower in areas where crocodile populations are smaller, where human water use is more limited, or where physical and behavioral modification of high-risk situations has been successfully implemented. They are higher during the breeding season, when crocodilians are more actively patrolling territories and are more likely to be encountered in marginal habitats near human settlements. And they are higher at dawn and dusk, when crocodilians are most actively hunting and when human visibility is reduced. The communities most exposed to crocodile attack risk are also typically the communities with the least economic and institutional capacity to manage that risk. Rural communities in sub-Saharan Africa, Southeast Asia, and Northern Australia, whose livelihoods depend on waterway access and who cannot easily afford or access the protective infrastructure that reduces risk. This intersection of conservation-relevant predator recovery and disproportionate risk burden on economically marginalized communities is one of the most challenging dimensions of crocodilian conservation policy, and it requires responses that are sensitive to both the ecological value of the predator and the legitimate safety and livelihood interests of the communities most affected. Programs that have successfully navigated this challenge share several characteristics. They involve genuine community participation in both risk management planning and in the benefits of crocodile conservation through wildlife tourism revenue sharing, community employment and conservation monitoring programs, and meaningful consultation and management decisions. They are supported by government institutions with both the mandate and the resources to implement protective infrastructure at high-risk locations. They are accompanied by sustained culturally appropriate public education about crocodile behavior and risk reduction that is developed with community input rather than simply broadcast from outside. And they maintain honest, transparent communication about residual risk, acknowledging that even with optimal management, some risk remains in areas where large crocodilians and active human populations share waterways, rather than making promises about safety that cannot be kept, and that, when broken by an attack, undermine public trust in conservation programs. The Nile crocodile's situation across its African range illustrates both the successes and the ongoing challenges of crocodilian conservation in a developing world context. Crocodilus niloticus was heavily hunted through the mid-20th century, with populations severely reduced across much of its range by commercial skin hunters. Legal protections implemented in most African countries through the 1970s and 1980s, combined with the listing of the species under CITES Appendix I, prohibiting commercial trade, except from populations specifically authorized for sustainable use allowed most populations to begin recovering by the late 1980s and 1990s. Recoveries in some areas, the Nile system in southern Sudan, the Zambezi drainage, the Kafua and Luangwa systems in Zambia, were substantial enough that population managers began to consider whether sustainable use programs allowing limited commercial harvest of skins and other products could be implemented in ways that maintain population health while generating economic incentives for local communities to support continued conservation. The sustainable use debate in crocodilian conservation mirrors debates across wildlife management more broadly, and it involves genuine scientific and ethical complexity that resists simple resolution. On the scientific side, the question is whether populations are sufficiently large, the harvest rates sufficiently low, and the monitoring and enforcement systems sufficiently robust to ensure that legal harvest does not drive population decline. On the ethical side, the question is whether the commercial exploitation of a wild animal for luxury products, crocodile leather, remains among the most expensive animal-derived materials in the fashion industry, is compatible with conservation ethics, and whether the economic benefits of that exploitation accrue to the communities and institutions whose conservation support is most needed, or are captured by commercial interests with less stake in long-term population health. The CITES-managed ranching and farming programs that have been developed for several crocodilian species, most extensively for Crocodilus niloticus in southern and eastern Africa, and for Crocodilus porus in Australia, Papua New Guinea, and parts of Southeast Asia, represent an attempt to navigate this complexity by creating regulated commercial use systems with sufficient monitoring and enforcement to maintain wild population health. These systems have had mixed, but generally positive outcomes for wild populations in areas where they have been effectively implemented, and they have generated meaningful economic incentives for community conservation support. In some areas, their failure modes occur when enforcement is inadequate, when the commercial interests operating within the system create pressure for harvest rates that exceed sustainable levels, or when the distribution of economic benefits is so skewed toward commercial operators that communities receive insufficient incentive to maintain conservation behaviors. The gharial Gavialis gangeticus presents the starkest conservation failure story in the crocodilian order, and it deserves careful examination because it illustrates the specific vulnerabilities of highly specialized species to habitat modification and the difficulty of recovering a population that has declined below certain demographic thresholds. The gharial is, by most measurements, the most ecologically specialized living crocodilian, a fish specialist whose extremely long, narrow snout, lined with interlocking needle-like teeth, is optimized for the rapid lateral sweeping motion used to catch fish in river currents. This specialization makes it one of the most effective fish predators in the rivers it inhabits, but it also makes it dependent on specific river conditions. Clear, fast-flowing water with sandy banks for nesting that have been dramatically modified across its former range in the river systems of the Indian subcontinent. The gharial's range historically extended across the river systems of the entire Indian subcontinent, the Indus, Ganges, Brahmaputra, Mahanadi, and their major tributaries. By the 1970s, hunting, egg collection, and habitat modification had reduced the population to an estimated 200 individuals, confined to a few remnant stretches of suitable river habitat in India and Nepal. Conservation programs launched in the late 1970s, involving captive breeding and release of hatchlings into protected river stretches, produced apparent population recoveries over the following 2 decades, with estimates reaching several thousand individuals by the early 2000s. Then, in 2007 and 2008, a catastrophic mortality event killed large numbers of adult gharials in the Chambal River, one of the last strongholds of the species, in ways that initial investigations suggested were related to gout-like organ failure, possibly associated with pollution or prey contamination. The causes were never definitively established, and the population has not recovered to pre-2007 levels. The current wild gharial population is estimated at fewer than 1,000 individuals, making it one of the most critically endangered crocodilians and one of the most critically endangered large vertebrates on Earth. Its situation illustrates the precariousness of populations that have been reduced below certain thresholds, where demographic stochasticity, reduced genetic diversity, accumulated environmental stressors, and the continued pressure of habitat modification combine to make recovery extremely difficult, even with significant conservation investment. The recovery of the gharial, if it happens, will require not just the protection of the animals themselves, but the restoration of river conditions, flow regimes, water quality, sand bank availability that have been altered by decades of damming, sand mining, agricultural runoff, and pollution in ways that no simple legal protection can reverse. The false gharial Tomistoma schlegania of Southeast Asia occupies a taxonomic position that has been the subject of scientific controversy for decades. Morphologically, it resembles the gharial in its long, narrow snout, and it was long classified alongside the true gharial in family Gavialidae on this basis. Molecular genetic analysis has produced conflicting results, some analyses placing Tomistoma within Gavialidae, others suggesting it is more closely related to the true crocodiles of family Crocodilidae. The most recent analyses using the most complete genetic data sets favor placement within Gavialidae, but the question is not definitively settled. Whatever its precise phylogenetic position, Tomistoma is a large, semi-aquatic predator of the peat swamp forests and large rivers of Borneo, Sumatra, peninsular Malaysia, and Java, and it is severely threatened by habitat loss driven by the conversion of peat swamp forests to oil palm plantations, one of the most rapid and extensive habitat transformations occurring anywhere on Earth. Its conservation status is vulnerable, but this may understate the urgency of its situation given the rate at which its peat swamp habitat is being eliminated. The caimans of Central and South America represent the most diverse group of living crocodilians, with 6 species occupying habitats ranging from the vast flooded savannas of the Pantanal to the blackwater rivers of the Amazon basin to the high-altitude lakes of the Andes. The spectacled caiman, Caiman crocodilis, is the most abundant crocodilian on Earth, with a population estimated in the millions across its range. And the black caiman, Melanosuchus niger, is the largest predator in the Amazon basin, historically growing to 5 meters or more and occupying the apex predator role in the most biodiverse river system on the planet. The black caiman was hunted to near extinction by commercial skin hunters during the mid-20th century. Its range-wide population may have been reduced by 95% or more at the nadir of hunting pressure and has partially recovered following legal protection, but it remains far below historical abundances across much of its Amazon range. The Cuvier's dwarf caiman, Paleosuchus palpebrosus, deserves mention as the other extreme of crocodilian size variation at maximum lengths of 1.6 meters. It is the smallest living crocodilian, adapted to fast-flowing rocky streams and rivers in the Amazon and Orinoco basins where larger crocodilians cannot effectively operate. Its heavily ossified skin, the most heavily armored of any living crocodilian, provides protection in an environment where the rocks and strong currents impose physical stresses that the softer skin of larger species could not withstand. The Cuvier's dwarf caiman is little studied and poorly understood by comparison with larger, more commercially significant species, and it represents the kind of ecologically important but scientifically underserved organism whose conservation status and ecological role remain incompletely characterized. The American alligator, alligator mississippiensis, occupies a special place in the history of crocodilian conservation because its recovery from near extinction in the mid-20th century to current population estimates of approximately 5 million individuals in the southeastern United States represents one of the most dramatic and well-documented wildlife recovery stories in American conservation history. By the 1950s and 1960s, decades of unregulated hunting for skins and meat had reduced alligator populations across the southeastern states to a fraction of their historical abundance. With the species listed as endangered in 1967, federal protection under the Endangered Species Act, combined with state-level management programs and the cessation of commercial hunting, allowed populations to recover rapidly, a reflection of the alligator's biological resilience and its tolerance for a wide range of habitat conditions across the southeastern coastal plain. By 1987, the alligator had recovered sufficiently to be removed from the endangered species list, and today it is managed as a game species in several states, with regulated hunting seasons generating economic value from a recovered population. The alligator's recovery transformed the wetland ecosystems of the southeastern United States in ways that are now extensively studied. Alligator holes, depressions excavated by alligators in wetland substrates, which fill with water and maintain water levels during dry periods, are recognized as critical habitat features for dozens of species, including fish, amphibians, invertebrates, and birds that depend on these water sources during droughts. The alligator is a textbook example of a keystone species whose engineering activities create habitat for a broader community of organisms, and whose removal from an ecosystem causes measurable changes in community structure that go far beyond the simple removal of one predator. The Chinese alligator, Alligator sinensis, is the alligator's only congener, a small, critically endangered species restricted to a tiny portion of its historical range in the lower Yangtze River Delta of eastern China. Once ranging across much of eastern China, in the lowland river systems of the Yangtze drainage, it has been reduced by habitat loss, the conversion of its wetland habitat to agriculture over millennia of intensive land use, and by direct persecution to a wild population estimated at fewer than 150 individuals. Confined to a few fragmented wetland remnants in Anhui Province, it is maintained in larger numbers in captive breeding programs, both in China and in American zoos. And reintroduction efforts are underway, but the availability of suitable, adequately protected habitat for reintroduction remains a limiting constraint that makes the long-term recovery of the wild population uncertain. The contrast between the American alligator, 5 million individuals managed as a game species, and the Chinese alligator, fewer than 150 wild individuals, critically endangered, illustrates a pattern visible across the crocodilian order, that the conservation status of individual species reflects not simply their biological characteristics, but the specific history of human land use, hunting, and policy response in the regions they inhabit. Biologically, the two alligator species are closely related and not dramatically different in their ecological requirements or resilience. The American alligator recovered because hunting was stopped in time, adequate habitat remained, and an effective regulatory framework was established and enforced. The Chinese alligator did not recover, or rather has not yet recovered, because the combination of habitat loss and human persecution was more severe and longer lasting, and because the habitat conditions required for wild population recovery are harder to create in one of the most densely populated and intensively cultivated landscapes on Earth. This lesson that the outcome for any given crocodilian population depends critically on the specific human decisions made about hunting, habitat use, and wildlife management, and that different outcomes for biologically similar species reflect different human choices rather than different biological destinies, is perhaps the most important general insight that the crocodilian order offers for conservation science. These animals are biologically resilient. They have demonstrated repeatedly the capacity to recover when direct threats are reduced and adequate habitat remains. The question is always whether human societies will make the choices, in time, at adequate scale, with adequate enforcement, that allow that biological resilience to express itself. The biology of crocodilian reproduction is one of the most sophisticated and ecologically consequential aspects of their life history, and it reveals layers of physiological and behavioral complexity that continue to surprise researchers who approach these animals with the assumptions inherited from older, simpler models of reptilian biology. Reproduction in crocodilians is not a simple matter of laying eggs and hoping for the best. It is an extended, energetically costly, behaviorally complex process that involves territory establishment, mate, selection, nest construction, incubation management, hatchling care, and extended maternal guarding, a reproductive investment that, per breeding attempt, rivals or exceeds that of many mammals and birds. The mating systems of crocodilians vary among species, but most are best described as polygynous, dominant males establishing territories that overlap with those of multiple females, with mating access determined by a combination of size-based dominance and behavioral display. The bellowing and infrasound displays of male alligators during the breeding season, the famous alligator water dance, in which the vibrations produced by the vocalizing male cause the water surface around the animal to leap in a halo of droplets, are visual and acoustic demonstrations of size and vigor that communicate competitive ability to both rival males and potential mates. Similar display behaviors occur across the crocodilian order, varying in specific form among species, but sharing the common function of communicating competitive status without requiring the energetically costly and physically risky engagement of direct combat. Direct combat does occur, particularly among males of similar size competing for access to females or to high-quality territories. These encounters can be violent and prolonged, involving the use of the full force of the jaws and the torsional power of the body in ways that regularly produce serious injuries, lost limbs, severe lacerations, broken jaws, the scarring visible on the bodies of large adult male crocodilians in any wild population is the accumulated physical record of these encounters, and it tells a story of competitive intensity that contradicts any simple image of the crocodilian as a slow, passive animal, the animal that waits motionless in the shallows for hours, expending minimal energy in patient ambush, is the same animal that engages in explosive full-contact territorial combat when competitive stakes demand it. This behavioral range, from absolute stillness to explosive, violent action, with no apparent and intermediate states, is one of the defining characteristics of the crocodilian behavioral repertoire. Female nest site selection is not random. Long-term studies of nesting populations of multiple crocodilian species have shown that females return repeatedly to the same nesting areas year after year, and that nest site characteristics, proximity to water, soil temperature and moisture, degree of shading, distance from other nests, and from potential predator access routes, are assessed with a precision that implies sensory evaluation of multiple environmental parameters. The nest itself, in mound nesting species like the Nile crocodile and the saltwater crocodile, is not simply a pile of vegetation. It is a carefully constructed thermal environment designed to maintain the eggs within the temperature range required for successful incubation, which in crocodilians is approximately 28 to 32 degrees Celsius, depending on species. The decomposition of the organic material in the nest mound generates heat, supplementing solar warming, and the female's behavioral management of nest temperature, opening and closing the nest, adding or removing material, shading the nest with her body, is an active thermoregulatory intervention that maintains conditions within the required range through the considerable temperature fluctuations of a tropical wet season. Temperature during incubation does more than simply determine whether eggs develop successfully. In crocodilians, as in many other reptiles, the sex of the developing embryo is determined not by genetic sex chromosomes, as in mammals and birds, but by the temperature experienced during a critical period of embryonic development, a phenomenon called temperature-dependent sex determination, or TSD. In most crocodilian species, intermediate incubation temperatures produce females, while both higher and lower temperatures produce males, a pattern opposite to that seen in many turtle species, and one whose ecological logic is not fully understood. What is clear is that the precise temperature management of the nest by the attending female has direct consequences for the sex ratio of the offspring she produces, and that this mechanism of sex determination creates a vulnerability to climate change that is only beginning to be appreciated in its full implications. As global temperatures rise, the thermal environments experienced by crocodilian nests will shift. In species where specific temperature ranges determine offspring sex, population-level sex ratio distortions could result, potentially skewing populations toward one sex and reducing reproductive efficiency below viable levels. This concern is not hypothetical. It has already been documented in some turtle populations experiencing sex ratio skews toward females as nesting beach temperatures have risen. Whether and to what degree similar effects will manifest in crocodilian populations depends on species-specific TSD reaction norms. The degree to which female nest site selection behavior can track shifting thermal environments and the pace of temperature change relative to the generation time of these long-lived animals. These are questions that current research is actively addressing, and the answers will be important for anticipating which crocodilian populations are most vulnerable to climate-related reproductive disruption. The longevity of crocodilians is another dimension of their biology that has significant conservation implications, and that is incompletely characterized, even for well-studied species. Crocodilians are long-lived animals. Estimates of maximum lifespan for large species like Crocodilus porosus and Crocodilus niloticus range from 60 to 100 years or more, though the evidence for extreme longevity in wild individuals is difficult to obtain because tracking individual animals over such timescales requires sustained monitoring programs that have only recently become available. What is well established is that large crocodilians reach sexual maturity relatively late. Females of most large species do not begin breeding until they are 7 to 10 years old, and males later still, and that they continue to grow throughout their lives, a pattern called indeterminate growth. That means the largest individuals in any population are also typically the oldest and most reproductively experienced. This life history pattern, late maturity, long lifespan, indeterminate growth, has several important conservation implications. It means that the largest individuals in a population, which are preferentially targeted by both commercial hunters and trophy hunters because of the high value of their large skins and the cultural significance of large trophies, are also the most reproductively important. The most experienced breeders, the holders of the highest quality territories, the animals most likely to produce surviving offspring, selectively removing the largest individuals from a population therefore disproportionately impairs reproductive output and can cause population-level demographic changes that are not immediately apparent from population size alone. It also means that populations recovering from hunting pressure require long periods, potentially decades, to restore the full size class distribution that characterizes an unexploited population, even if hunting has ceased and population numbers are growing. The genetics of crocodilian populations has been investigated with increasing sophistication over the past 2 decades, and the picture that has emerged is more complex and more interesting than earlier models suggested. Crocodilians show meaningful genetic structure across their ranges, with populations in different river systems or geographic regions often showing genetic differentiation that reflects the limited dispersal of individuals between watersheds separated by terrestrial habitats inhospitable to crocodilians. At the same time, the capacity of saltwater crocodiles and some other species for long-distance ocean or river dispersal means that gene flow between some geographically distant populations is higher than expected, and that the genetic units relevant for conservation management do not always coincide with the administrative boundaries, national borders, state lines, protected area perimeters within which conservation programs operate. The identification of distinct genetic lineages within broadly distributed species like Crocodilus niloticus has challenged the taxonomic assumptions underlying conservation policy. Recent genetic analyses suggest that the Nile crocodile, as traditionally defined, may actually comprise 2 or more distinct species, a West African lineage, Crocodilus successa, which was recently elevated to full species status, and the Nile crocodile sensu stricto, Crocodilus niloticus, of eastern and southern Africa. If this split is confirmed and broadly accepted, it changes the conservation status of both lineages. The smaller, more restricted West African population would warrant higher conservation concern as an independent unit than it receives as part of the broader Nile crocodile complex. This taxonomic revision illustrates the general principle that the units we choose to recognize and protect in conservation policy, species, subspecies, distinct population segments reflect both biological reality and human decisions about how to parse that reality, and that advances in genetic methodology regularly challenge and refine those decisions. The ecological interactions between crocodilians and other apex predators in shared habitats are complex and ecologically significant in ways that are only beginning to be systematically studied. In African river systems, Nile crocodiles share the apex predator role with hippopotami and with large terrestrial predators. Lions, leopards, hyenas, wild dogs that come to water to drink and occasionally to hunt. The interactions between crocodiles and hippos are particularly interesting because they represent the coexistence of 2 large, potentially competitive species that partition the apex role along ecological lines. Hippos dominating the aquatic vegetation, and terrestrial grazing resource base. Crocodiles dominating the aquatic predation niche, without the explosive competitive displacement that would be expected if their ecological roles overlapped more fully. In the rivers of South Asia and Southeast Asia, saltwater and mugger crocodiles share habitats with large constrictor snakes, pythons in particular, creating predator assemblages of unusual diversity and competitive complexity. Documented interactions between these species reveal a degree of competition and occasional predation in both directions. Large pythons have been documented killing juvenile crocodilians, while large crocodilians readily prey on pythons. That suggests these co-occurring apex predators have a more dynamic and interactively structured relationship than the simple image of a stable apex predator community implies. In Florida and increasingly across the southeastern United States, the introduced Burmese python has created a novel competitive interaction with the American alligator in the Everglades ecosystem, with documented cases of large pythons successfully constricting and consuming juvenile alligators, and of alligators successfully killing and consuming pythons. This interaction is playing out in an ecosystem that evolved without large constricting snakes, and its long-term consequences for the alligator population and for the broader Everglades food web are a subject of active research and management concern. The python invasion illustrates the vulnerability of native predator communities to disruption by introduced species, a vulnerability that is relevant for crocodilian conservation in multiple parts of the world where the introduction of non-native predators or prey species is altering the ecological context in which crocodilian populations operate. The relationship between crocodilians and fish communities is one of the most ecologically important and least studied dimensions of crocodilian ecology. Crocodilians consume fish at all life stages, from the small fish taken by hatchlings and juveniles to the large catfish, cichlids, and other fish taken by adults. And the cumulative predation pressure of a large crocodilian population on the fish communities of a river system can be substantial. At the same time, crocodilians contribute to fish production through the nutrient cycling associated with their kills, through the creation of deepwater refugia in alligator holes, and through the behavioral effects of predation risk on fish movement and habitat use that alter competitive interactions among fish species in ways that generally maintain diversity. In the Pantanal, the world's largest tropical wetland, the Yakar caiman yakar, reaches densities of several thousand individuals per square kilometer in some areas, among the highest densities recorded for any large predator anywhere on Earth. At these densities, the caimans are major consumers of pyron, the abundant and ecologically important predatory fish of the Pantanal River system. Research has shown that caiman predation maintains piranha populations below the levels at which their own predatory impact on other fish species would become destabilizing, a top-down regulation that cascades through multiple trophic levels and that helps maintain the extraordinary fish diversity for which the Pantanal is renowned. This is a textbook trophic cascade operating in real time in one of the world's most biodiverse freshwater ecosystems, and the caiman, often overlooked in favor of more charismatic Pantanal megafauna like jaguars and giant otters, is the functional engine driving it. The biomedical significance of crocodilians has emerged as an unexpected but scientifically compelling dimension of research interest. Driven by observations of the extraordinary resistance of these animals to infection, blood loss, and physical trauma, crocodilians routinely suffer severe wounds from territorial combat, from encounters with prey, from interactions with human infrastructure, that would be quickly lethal in most other vertebrates through blood loss, shock, or infection. Instead, they heal with a speed and completeness that researchers have found remarkable. Analysis of crocodilian blood plasma has identified compounds with potent antimicrobial activity, including activity against bacteria that are resistant to conventional antibiotics, such as methicillin-resistant Staphylococcus aureus. The molecular mechanisms underlying this antimicrobial activity are subjects of active investigation, with implications for the development of novel antimicrobial compounds at a time when antibiotic resistance is one of the most serious emerging threats to global public health. The blood of crocodilians also contains unusual hemoglobin variants and oxygen transport mechanisms that allow the animal to sustain prolonged dives, up to 2 hours in some documented cases, without the neurological damage that oxygen deprivation causes in most other vertebrates. Understanding the molecular basis of this hypoxia tolerance has potential biomedical applications in the contexts of stroke, cardiac surgery, and organ transplantation, where the ability to tolerate temporary oxygen deprivation without tissue damage is clinically valuable. These potential applications are speculative at the current state of research, but they have attracted serious scientific attention and have generated a stream of publications examining crocodilian physiology from a biomedical perspective that complements the ecological and evolutionary research that has dominated the field. The skin regeneration capacity of crocodilians is another biomedically relevant trait. The speed and efficiency with which these animals heal large wounds, restoring the complex, multi-layered structure of the skin, including the osteoderms, with a completeness that resembles the original tissue more closely than the scar tissue produced by mammalian wound healing, is being studied as a potential model for understanding the cellular and molecular mechanisms that could be harnessed to improve wound healing in humans. The comparison is imperfect. Crocodilian and mammalian skin are structurally and biochemically different in important ways. But the fundamental question of how one vertebrate lineage evolved such dramatically enhanced regenerative capacity compared to another is one that developmental biologists find intrinsically interesting, independent of its clinical applications. The cultural relationship between crocodilians and human societies extends back as far as human culture itself, and across every continent where crocodilians live, the archaeological, artistic, and ethnographic record documents a relationship of extraordinary depth and complexity. In ancient Egypt, the crocodile god Sobek was one of the most widely worshipped deities of the Egyptian pantheon, associated with the fertility of the Nile flood, with royal power, with the protection of waterways, and with the primordial creative force that brought order from chaos at the beginning of the world. Sobek temples at Kamambo and at Fayum maintained living sacred crocodiles, animals kept in pools within the temple precinct, fed by priests, adorned with jewelry, and treated as living manifestations of the divine. The mummified remains of thousands of crocodiles, ranging from hatchlings to large adults, have been recovered from ancient Egyptian burial sites, representing offerings, votive objects, and the mortal remains of sacred animals, whose significance to the ancient Egyptians was profound and multidimensional. In the indigenous cultures of Australia, the crocodile occupies a central position in the Dreamtime narratives of many Aboriginal peoples of the northern regions where saltwater and freshwater crocodiles are abundant. These narratives encode ecological knowledge about the behavior, habitat preferences, and seasonal patterns of crocodilians, within cultural and spiritual frameworks that have served the dual function of maintaining knowledge across generations and investing the natural world with moral and spiritual significance that motivates respectful and sustainable interaction. The complexity and ecological accuracy of this traditional knowledge, when examined by researchers with sufficient cross-cultural competence to understand it in its proper context, has repeatedly revealed insights about crocodilian behavior and ecology that formal scientific observation has subsequently confirmed. A reminder that traditional ecological knowledge developed through millennia of close observation by people whose survival depended on accurate knowledge of their environment represents a scientific resource of genuine value. In the cultures of Central America, the caiman appears throughout the iconography of pre-Columbian civilizations from the Olmec through the Maya and Aztec, associated with the earth, with water, with primordial creative power, and with the underworld. The Aztec earth deity Tlaltecuhtli was frequently depicted in crocodilian form, a giant caiman-like creature floating on the primordial waters, whose body was transformed by the gods to create the surface of the earth. In the cosmologies of many Amazonian indigenous peoples, the caiman is a transformer figure, a being whose existence at the boundary between the aquatic underworld and the terrestrial surface world makes it a liminal entity of special spiritual significance, capable of moving between the realms that human beings must navigate in their social and spiritual lives. These cultural relationships are not simply interesting historical curiosities. They are living realities in many communities across the crocodilians range today, and they have direct implications for conservation programs that must operate within these cultural contexts. In communities where the crocodilian is a sacred or spiritually significant animal, conservation can build on existing cultural values that support the animal's protection. In communities where the crocodilian is feared, associated with malevolent supernatural forces, or viewed as a manifestation of the dangers of the natural world against which human communities must defend themselves, conservation programs face cultural headwinds that must be addressed through culturally sensitive engagement rather than simply overridden by external authority. The contemporary situation of crocodilians globally reflects the intersection of all these threads: evolutionary history, ecological function, physiological sophistication, cultural significance, conservation history, and the ongoing challenges of human-wildlife coexistence in a rapidly changing world. The picture is mixed, with genuine successes in some regions and species alongside ongoing decline and crisis in others, and with the overarching pressure of climate change and habitat modification creating new challenges that add to rather than replace the older threats of hunting and direct persecution. Of the 25 living crocodilian species, 6 are currently listed as critically endangered by the IUCN: the gharial, the Philippine crocodile, the Siamese crocodile, the Cuban crocodile, the Orinoco crocodile, and the Chinese alligator. 3 are listed as vulnerable, and several others are listed as data deficient, meaning that insufficient information exists to assess their conservation status with confidence. The more abundant species, the spectacled caiman, the American alligator, the saltwater crocodile, have stable or increasing populations in most parts of their range, while the rarer, more habitat-specialized, or more heavily hunted species remain at serious risk. The Philippine crocodile, Crocodiles mindorensis, is one of the least known and most threatened of the critically endangered species. Endemic to the Philippines, it has been reduced to a wild population estimated at fewer than 150 mature individuals by a combination of hunting, habitat loss, and human-wildlife conflict. Its restricted range, several isolated freshwater populations on the islands of Luzon and Mindanao, makes it vulnerable to the catastrophic weather events, disease outbreaks, and local habitat changes that can eliminate small, isolated populations. Conservation programs led by the Mabuhay Foundation and other Philippine conservation organizations have achieved some successes in increasing local community support for the species and in documenting population trends. But the overall situation remains precarious. The Siamese crocodile, Crocodile siamensis, was believed extinct in the wild by many researchers until surveys in the late 90s and 2000s documented small surviving populations in Cambodia, Laos, Vietnam, and Borneo. The Cambodian population in the Cardamom Mountains is the largest known wild population and has been the focus of intensive conservation work by the Fauna and Flora International and partner organizations involving community-based monitoring, nest protection, and captive breeding and release programs. The Siamese crocodile faces threats from habitat loss, hunting, and hybridization with the more abundant saltwater crocodile, which has produced hybrid individuals in some areas whose genetic status complicates population management. Despite these challenges, the Cambodian population appears to be stable or slowly increasing under active management, providing cautious grounds for optimism about the species' ability to recover given adequate protection. The Orinoco crocodile, Crocodile intermedius, historically the apex aquatic predator of the Orinoco Basin of Venezuela and Colombia, is one of the most severely depleted crocodilians in the Americas. Commercial hunting for skins during the mid-20th century reduced the population to an estimated 250 individuals at its nadir, one of the lowest population sizes ever recorded for a large vertebrate prior to a conservation intervention. Protection and captive breeding programs have allowed partial recovery, with current estimates suggesting a wild population of perhaps 1,500 individuals. But this remains far below historical levels, and the species is considered to be at high risk of extinction without continued and expanded conservation effort. The Orinoco crocodile's recovery is complicated by the political instability of Venezuela, which has disrupted conservation programs, reduced enforcement of wildlife protection laws, and created conditions in which illegal hunting has resumed in some areas. The Cuban crocodile, Crocodiles rhambifer, occupies the smallest natural range of any living crocodilian, the Zapata swamp and Lanier swamp of Cuba, and faces a specific threat that makes its conservation situation unusually complex: hybridization with the American crocodile, Crocodilus acutus, which also occurs in Cuba. As the Cuban crocodile population has declined and the American crocodile has been reintroduced to or recovered in overlapping areas, the two species have interbred, producing hybrid individuals that are genetically intermediate. If this hybridization continues unchecked, the genetic integrity of the Cuban crocodile as a distinct species, with its unique adaptations to the specific conditions of the Zapata swamp, will be progressively diluted. Managing this hybrid threat requires identifying purebred Cuban crocodiles through genetic analysis and implementing management decisions about hybridized individuals that are scientifically complex and ethically contentious. The conservation of crocodilians in the 21st century faces challenges that are fundamentally different in character from those that dominated the 20th century conservation response. The 20th century crisis was primarily one of direct exploitation, commercial hunting driving populations below viable levels, and the primary conservation response was regulatory, banning hunting, establishing legal protections, creating reserves, This response worked for the species and populations that received it in time. The 21st century crisis is more diffuse, more systemic, and less amenable to simple regulatory solutions. It involves the modification of entire hydrological systems through damming, water extraction, and channel engineering that changes the conditions crocodilians depend on for nesting and thermoregulation. It involves the contamination of river systems with agricultural, industrial, and pharmaceutical pollutants that accumulate in crocodilian tissues and impair reproduction and immune function. It involves the fragmentation of wetland habitats by infrastructure development that isolates populations and impairs the dispersal that maintains genetic diversity. And it involves the climate-driven changes in temperature, precipitation patterns, and extreme weather events that alter the conditions experienced by nesting eggs, by hatchlings, and by the prey communities on which crocodilians depend. Addressing these systemic drivers requires engagement with economic systems, planning processes, and policy frameworks far beyond the traditional perimeter of wildlife conservation. Engagement with dam licensing authorities, agricultural policymakers, urban planners, pharmaceutical regulators. This broader engagement is both necessary and challenging for conservation organizations whose expertise and institutional relationships have historically been focused within the narrower domain of species protection and habitat management. Building the interdisciplinary collaborations and the policy engagement capacity that this broader challenge requires is one of the most important institutional developments currently underway in crocodilian conservation. The role of technology in 21st century crocodilian research and conservation is expanding rapidly, with developments that are transforming both the scientific understanding of these animals and the practical capacity to protect them. Satellite tracking, which has been applied to crocodilians for 2 decades, but whose resolution and data transmission capabilities have improved dramatically, is now producing movement datasets of a richness and temporal resolution that allow reconstruction of detailed individual behavioral histories over periods of years. Drone-based surveys, which can cover large areas of inaccessible wetland habitat in hours and detect crocodilians through both visual observation and thermal imaging, are replacing the labor-intensive boat and foot surveys that previously constrained the speed and cost of population assessments. Environmental DNA analysis, the detection of crocodilian DNA shed into the water column as skin cells, feces, and other biological material, is beginning to be applied as a non-invasive survey tool that can detect crocodilian presence in water bodies without any need to directly observe the animals. Acoustic monitoring, the deployment of underwater hydrophones to detect the infrasound and low-frequency vocalizations that crocodilians produce as part of their social and reproductive communication, is revealing the extent and complexity of crocodilian acoustic behavior in ways that direct observation cannot capture. Crocodilians are far more vocal than their surface behavior suggests, producing complex sequences of calls, rumbles, and vibrations that other individuals respond to, in ways indicating that these vocalizations carry meaningful information about identity, location, and behavioral state. The study of this acoustic communication system is in its early stages, but initial results suggest that it is considerably richer and more informative than earlier models of crocodilian social behavior recognized. Artificial intelligence and machine learning are being applied to the analysis of camera trap and drone imagery, automating the detection and counting of crocodilians in survey images and enabling the processing of data sets too large for manual analysis. individual identification of crocodilians from photographic records, analogous to the spot pattern identification used for snow leopards and the stripe pattern identification used for tigers, is being developed using AI-based image analysis tools that can match individuals across thousands of photographs from multiple camera stations, enabling mark recapture population estimation at scales previously impossible. These tools are not fully mature, and their validation requires careful comparison with results from established methods, but their potential to dramatically reduce the cost and increase the efficiency of crocodilian population monitoring is significant. The integration of crocodilian conservation with broader freshwater ecosystem conservation is increasingly recognized as both ecologically necessary and strategically advantageous. Freshwater ecosystems are among the most threatened on Earth. Freshwater biodiversity is declining faster than either marine or terrestrial biodiversity, driven by the combined pressures of water extraction, pollution, dam construction, and invasive species. The crocodilian, as the apex predator of many freshwater systems, is both a sensitive indicator of freshwater ecosystem health and a flagship species whose conservation requirements align closely with the requirements of the broader freshwater biodiversity that shares its habitat. Conservation programs that frame crocodilian protection within the broader context of freshwater ecosystem protection, making the case that healthy rivers, lakes, and wetlands support both crocodilians and the biodiversity that makes those systems valuable to human communities, are more likely to generate the broad-based political and economic support that effective freshwater conservation requires than programs focused narrowly on the crocodilian itself. The economic value of crocodilians to human communities extends well beyond the commercial value of their skins and meat. Important as those economic flows are in some contexts, crocodilians attract wildlife tourists who generate significant economic activity in areas where they can be reliably observed, from the crocodile watching cruises of the Northern Territory of Australia to the nighttime spotlighting tours of the Florida Everglades, from the river safaris of Southern Africa to the community-based crocodile tourism programs emerging in parts of Southeast Asia. In some ecosystems, the fish production that crocodilian predation supports through the trophic cascade effects described earlier is a direct economic benefit to fishing communities that depend on those fish for livelihood and food security. And the biomedical research value of crocodilian physiology, while not yet translated into commercial products, represents a potential economic benefit of uncertain but possibly substantial magnitude. The communities that live alongside large crocodilian populations and bear the costs of that coexistence in the form of livestock predation, attack risk, and restrictions on water use deserve fair compensation for those costs from the broader society that benefits from the conservation of these ecologically and economically valuable animals. This principle of equitable burden sharing in conservation, that the costs of maintaining globally or regionally valuable wildlife should not fall disproportionately on the local communities most exposed to those costs, is increasingly recognized in conservation policy but is still inadequately implemented in practice. Benefit-sharing mechanisms that channel a meaningful portion of the economic value generated by crocodilian conservation from tourism, from commercial use programs, from ecosystem service payments, to the communities that bear the highest costs are both ethically necessary and practically important for maintaining the community's support that long-term conservation requires. There is a particular kind of knowledge that only comes from years of watching the same animal in the same place across the full cycle of seasons, and the researchers who have accumulated that knowledge about crocodilians describe their subjects with a specificity and an intimacy that population-level statistics and physiological measurements cannot capture. They describe individual animals by name, or rather by the alphanumeric codes of research programs, though the names inevitably follow in field notes and in conversation. And they describe individual personalities, individual quirks, individual behavioral tendencies that persist across years of observation and that influence in measurable ways the ecological outcomes of each animal's life. This individual variation, the recognition that crocodilians are not interchangeable units in a population, but distinct individuals whose specific characteristics matter for their survival, reproduction, and ecological impact, is one of the most important conceptual advances in crocodilian research of the past 2 decades, and it has implications that extend well beyond crocodilian biology into the broader science of animal behavior and conservation management. The documentation of individual personality in crocodilians draws on a methodology developed originally for mammalian and avian behavioral research, in which individual animals are scored on standardized behavioral dimensions: boldness, exploration, aggression, sociability, reactivity to novelty, across multiple contexts and multiple time points. And the consistency of those scores across contexts and time is used as evidence for a stable individual trait rather than a situational response. Applied to crocodilians, this methodology has revealed consistent individual differences in approaches to novel objects, in response latency to feeding opportunities, in the degree of social tolerance toward conspecifics at shared resources, and in the willingness to engage with human observers at varying distances. These differences are not simply reflections of size or age, though both of those factors influence behavior. They are individual characteristics that persist when size and age are controlled for, and that predict with meaningful accuracy differences in hunting success, social rank, and reproductive outcomes. The existence of personality in crocodilians raises questions that go beyond the simple documentation of the phenomenon. If individual crocodilians have consistent behavioral tendencies that influence their ecological outcomes, then the removal of specific individuals from a population through targeted hunting, conflict management culling, or accidental mortality has consequences that cannot be predicted simply from population-level demographic models. Removing a bold, highly exploratory individual from a population may change the spatial dynamics of territory use in ways that affect multiple other individuals. Removing a particularly tolerant, socially sophisticated individual from a dominance hierarchy may trigger social instability and increased conflict that reduces the overall feeding efficiency of the group. These effects are real. They are beginning to be documented empirically, and they have management implications that population biologists and wildlife managers are only beginning to incorporate into their models and their decision-making frameworks. The question of individual recognition in crocodilians, whether these animals can distinguish specific other individuals from the same species, and if so, what information they use to do so, is closely related to the personality research and has been addressed through both field observation and controlled experiments. Field observations of crocodilians at shared feeding and basking sites have documented consistent dominance relationships between specific pairs of individuals that persist over years, suggesting that each animal maintains representations of specific others in its social world that allow it to calibrate its behavior appropriately to each individual encounter, rather than responding to every conspecific in the same way. controlled experiments in captive settings in which individuals are exposed to visual and acoustic stimuli from familiar and unfamiliar conspecifics have shown differential responses that indicate recognition of familiar individuals, responses that are more relaxed and less vigilant for familiar individuals, and more alert and reactive for unfamiliar ones. The mechanisms of individual recognition in crocodilians are not fully characterized, but the available evidence suggests that multiple sensory channels contribute. Visual recognition of body size, head shape, and the specific patterns of scarring and scale arrangement that make each individual visually distinctive is likely one component. Chemical recognition through olfactory and Jacobson's organ sensing of individual-specific scent profiles is another probable component. Given the importance of chemical communication in crocodilian social behavior, more broadly, acoustic recognition of individual-specific vocal characteristics, the specific frequency profiles and temporal patterns of bellows, hisses, and infrasonic rumbles that vary among individuals is a third probable component, particularly important for recognition at distances where visual and chemical signals are degraded. The integration of information from these multiple channels into a coherent individual identity representation requires neural processing of a complexity that earlier models of reptilian cognition would not have predicted, and it adds to the accumulating evidence for cognitive sophistication in these animals that the simple reptilian label has historically obscured. The role of learning in crocodilian predatory behavior deserves deeper examination than it has received in popular accounts of these animals, because it is directly relevant to understanding both the remarkable effectiveness of experienced predators and the particular vulnerability of naive individuals to human activities that more experienced animals have learned to avoid. Young crocodilians, in the period between independence from maternal care and the establishment of their own territories, are not equipped with a complete behavioral repertoire for their adult ecological role. They have innate predatory tendencies. The strike response to moving objects of appropriate size is present from hatching, but the specific techniques, the site preferences, the prey selection patterns, and the risk assessment that characterize the behavior of experienced adult hunters are products of learning accumulated over years of individual experience. This learning trajectory has been documented most clearly in long-term studies of Nile crocodiles at fixed observation sites where multiple generations of individuals have been monitored continuously. Juvenile crocodiles arriving at a productive feeding site for the first time show significantly lower hunting success than adults who have used the site for years. Not because their physical capacities are dramatically inferior, though they are smaller, but because they lack the site-specific knowledge that experienced individuals possess, the knowledge of which positions in the current field provide the best ambush angles, which approach routes are most likely to go undetected by prey, which prey species are most accessible, at which times of day, and how the behavior of prey changes in response to varying water levels, temperatures, and seasonal factors. This site-specific knowledge is acquired through experience, and it accumulates progressively over years of use of the same location, which explains the strong site fidelity that adult crocodilians show, returning year after year to the same productive locations rather than exploring for new ones. The implication of this learning trajectory for conservation management is significant and underappreciated. When crocodilians are relocated, moved from areas of human-wildlife conflict to protected areas or other locations deemed more suitable, they are moved away from the landscapes they know and into unfamiliar environments where their learned behavioral knowledge has no value. Relocated individuals frequently perform poorly in their new locations, failing to find productive feeding sites, establishing territories in suboptimal positions, and showing elevated stress indicators in physiological measurements. Some relocations result in the animal returning to its original location, sometimes covering extraordinary distances, a behavior that reflects the strength of the attachment to learned landscapes and that creates management complications when the original location is precisely the area of conflict that motivated the relocation. Understanding these learning dynamics changes how wildlife managers approach human crocodile conflict situations. Rather than defaulting to relocation as the standard response, moving the problem animal to a problem-free location, the most effective management approaches focus on modifying the human behavior or the physical environment that creates the conflict, leaving the crocodile in the landscape it knows and addressing the conditions that brought it into dangerous proximity to human activity. This approach is more demanding in terms of planning and community engagement than simple relocation, but it produces better outcomes for both the animal and the long-term management of the conflict. The aquatic environment that crocodilians inhabit presents sensory and navigational challenges that differ fundamentally from those of the terrestrial environment, and the solutions that crocodilians have evolved for those challenges reveal yet more layers of physiological and behavioral sophistication. Navigation in river systems, the ability to return to specific locations to locate productive feeding sites across large geographic areas, to find the way back to home territories after displacement, require spatial representations of the environment that are maintained across time and updated as the environment changes. The mechanisms of crocodilian spatial cognition and navigation are not yet fully characterized, but behavioral evidence for sophisticated navigation is compelling. Saltwater crocodiles that have been relocated by management authorities have been documented returning to their original territories across sea journeys of hundreds of kilometers. Journeys that require not simply heading in a consistent direction, but navigating around geographic obstacles through variable current regimes across open ocean where landmarks are absent to a specific destination whose location must be represented in the animal's memory with sufficient precision to guide the return. The sensory mechanisms that support this navigation are unknown. Magnetic field sensing, the use of the Earth's magnetic field as a navigational reference documented in sea turtles and some migratory birds, has been proposed as a component, and there is some indirect evidence consistent with this hypothesis, but direct demonstration of magnetic navigation in crocodilians has not yet been achieved. Whatever the mechanisms, the behavioral evidence for sophisticated spatial navigation in saltwater crocodiles represents one of the most striking demonstrations of cognitive capacity in any reptile, and it invites serious reconsideration of the cognitive complexity that the crocodilian body plan is capable of supporting. The thermal biology of crocodilians, the mechanisms by which they regulate body temperature in the absence of the internal heat generation that characterizes mammals and birds, is more sophisticated than the simple label of cold-blooded implies, and it has ecological consequences that are important for understanding how climate change will affect these animals. Crocodilians are behavioral thermoregulators. They use behavior, basking in sunlight, seeking shade, entering or leaving the water, to maintain body temperature within a preferred range that varies among species, but typically falls between 25 and 35 degrees Celsius for optimal physiological function. The effectiveness of behavioral thermoregulation depends on the availability of suitable basking sites, the thermal properties of the surrounding environment, and the energetic cost of the movements required to access different thermal environments. In stable climatic conditions, crocodilians are highly effective thermoregulators, maintaining body temperatures within their preferred range for the majority of daylight hours through a combination of behavioral adjustments that are choreographed with a precision that reflects the integration of real-time temperature sensing with behavioral response programs refined over millions of generations. The gaping behavior, lying with the mouth open for extended periods, that is characteristic of basking crocodilians, is partly a thermoregulatory behavior, exposing the highly vascularized mucous membranes of the mouth to evaporative cooling when body temperature approaches the upper preferred limit. The specific orientation of the body relative to the sun, the choice of basking substrate. Dark rock absorbs and retains heat more effectively than pale sand, and the timing of entries and exits from the water are all components of a thermoregulatory behavioral program of considerable sophistication. The disruption of this thermoregulatory system by climate change operates through multiple pathways. Rising ambient temperatures reduce the thermal gradient between the animal's preferred body temperature and the environmental temperature, which reduces the thermoregulatory benefit of basking and may push more of the active period into cooler nighttime hours, altering foraging and social behavior patterns in ways whose consequences for population ecology are not yet understood. Increased frequency and intensity of extreme heat events, heat waves that push environmental temperatures above the crocodilian's upper thermal tolerance, create conditions where behavioral thermoregulation alone may be insufficient to prevent physiological stress, requiring extended periods of immersion in cooler water that compete with time available for feeding, territorial maintenance, and reproduction. And changes in precipitation patterns that alter water levels and water temperatures in river systems and wetlands affect the thermal properties of the aquatic environments that crocodilians use as thermal buffers. These climate-related thermoregulatory challenges interact with the temperature-dependent sex determination system described earlier in ways that could produce population-level demographic changes that build, slowly remain invisible until they reach critical thresholds and are extremely difficult to reverse once those thresholds are crossed. A population whose sex ratio is progressively skewed toward one sex by warming nest temperatures does not immediately show signs of demographic distress. It may maintain stable adult population numbers for years or decades, while the productive adult sex ratio that drives population growth is quietly deteriorating. By the time the demographic consequences become visible in population trend data, the reproductive capacity of the population may be so compromised that recovery requires intervention at a level that was not anticipated when the problem was first detectable. This dynamic, the slow threshold crossing, difficult to reverse nature of climate-related demographic change, is one of the most challenging aspects of climate change for conservation management because it operates on timescales that exceed the monitoring periods of most conservation programs and produces effects that are only interpretable in retrospect. After the opportunity for preventive intervention has passed, addressing it requires proactive monitoring of nest temperatures and offspring sex ratios across multiple populations and multiple years. Monitoring that is technically demanding and expensive, and the integration of that monitoring data with demographic models capable of projecting population-level consequences before they become visible in adult population trends. The interaction between climate change and the disease ecology of crocodilians is another area of emerging concern that has received insufficient research attention. Crocodilians are hosts to a range of parasites and pathogens, bacteria, fungi, viruses, nematodes, trematodes, and others. And the dynamics of these host-pathogen interactions are sensitive to temperature in ways that are being altered by climate change. Some pathogens thrive at higher temperatures and may become more virulent or more transmissible as temperatures rise. Some parasites have complex life cycles involving intermediate hosts whose population dynamics are themselves altered by climate change, and whose changing abundance and distribution affects the exposure of crocodilians to infection. The mysterious mortality event that killed large numbers of gharials in the Chambal River in 2007 and 2008, whose causes were never definitively established, may have involved environmental conditions, water temperature, pollution load, prey availability, that were themselves influenced by climate variability, and whose recurrence under progressively warmer and more variable climatic conditions cannot be excluded. The immune system of crocodilians, while remarkable in its effectiveness against bacterial infection as described earlier, is not omnipotent. Large-scale captive breeding programs, which maintain high densities of crocodilians in conditions very different from their natural environment, have documented disease outbreaks, bacterial skin infections, respiratory illnesses, parasitic infestations that can cause significant mortality in captive populations and that represent management challenges for programs that depend on captive breeding for conservation and commercial purposes. The conditions of captive maintenance, crowding, artificial water quality, controlled feeding, create disease pressures that differ from those in the wild, and the immune systems of captive-bred individuals, never exposed to the full range of environmental pathogens encountered by wild individuals, may be less robustly primed than those of their wild counterparts, potentially compromising the survival of captive-bred individuals released into the wild. The reintroduction of captive-bred crocodilians into wild habitats is a conservation tool that has been applied with varying success across multiple species and regions. And the factors that determine reintroduction success are better understood now than when early programs were implemented but still incompletely characterized. Release site selection, ensuring that the habitat at the release location offers adequate food, appropriate thermal environments, suitable nesting conditions, and connectivity to existing wild populations, is critically important. The social context of release, whether individuals are released into areas already occupied by established wild crocodilians, and if so, how the social dynamics of resident populations respond to the addition of released individuals, is a factor that early programs often neglected, with consequences for release survival that subsequent research has helped to explain, and the life history stage at which individuals are released, hatchlings, juveniles, subadults, or adults, affects both survival rates and the behavioral competence of released individuals, with animals released at older, more experienced life stages generally showing better post-release survival than hatchlings released into the wild without any opportunity to develop the behavioral skills that wild experience provides. The most successful reintroduction programs have incorporated these lessons in ways that have improved outcomes measurably over the trajectory of program development. The Gharial Reintroduction Program in India, operating since the late 1970s and now spanning more than 4 decades of implementation, has evolved from early approaches that released large numbers of hatchlings with low survival expectations to more refined approaches that hold individuals in semi-wild conditions for 2 to 3 years before release, giving them time to develop predatory skills and behavioral competence while maintaining the logistical advantages of working with animals in a controlled setting. The survival rates of animals released under this improved protocol are substantially higher than those achieved under the earlier approach, though they remain far below what would be required to restore the wild population to self-sustaining abundance without continued supplementation from captive breeding. The Siamese crocodile reintroduction program in Cambodia has adopted a similar approach with the additional innovation of engaging local communities as active participants in the post-release monitoring of individuals, a strategy that simultaneously generates valuable data on release outcomes, builds community capacity and investment in the conservation program, and creates economic opportunities for community members whose alternative livelihood options in the remote Cardamom Mountains are extremely limited. Community monitors and trained by Fauna and Flora International staff to recognize and record individual crocodiles by the natural markings that distinguish them have generated monitoring data of a quality and continuity that professional research teams alone could not have achieved at the same cost. An example of citizen science in its most functional and genuinely participatory form. The legal frameworks governing crocodilian conservation and use are complex, multi-layered, and in some respects, internally inconsistent in ways that create both gaps in protection and perverse incentives that undermine conservation outcomes. CITES provides the primary framework for regulating cross-border trade in crocodilians and their products, with different species and populations listed in different appendices depending on their conservation status and the degree of trade control deemed appropriate. The mechanics of CITES listing and the criteria for moving species between appendices have been subjects of ongoing scientific and political negotiation since the convention entered into force in 1975, and the outcomes of these negotiations reflect not only the scientific evidence on population status and trade impacts, but also the political interests of range states, importing countries, and the commercial industries that profit from the trade. The sustainable use provisions of CITES, the mechanisms that allow regulated commercial trade in wildlife products from populations that can sustain that trade without population decline, have been applied to several crocodilian species in ways that have generated both positive conservation outcomes and ongoing controversy. The ranching programs for Nile crocodiles in Zimbabwe, South Africa, and several other southern African countries, which operate under CITES-approved quotas and generate revenue that is shared with local communities and conservation programs, have maintained wild Nile crocodile populations at healthy levels in the areas where they operate while generating the economic incentives for community conservation support that protection-only approaches cannot provide. Critics of these programs argue that they create demand for crocodilian leather products that cannot be fully met from legal, sustainable sources, thereby creating market conditions that stimulate illegal trade from non-sustainable sources, an argument that the evidence neither fully supports nor fully refutes, and that reflects genuine uncertainty about the complex market dynamics of legal and illegal wildlife trade. The national legal frameworks governing crocodilian conservation vary enormously among range countries, from comprehensive wildlife protection laws with effective enforcement in some developed country range states to minimal or unenforced protection in others. The gap between legal protection on paper and effective protection in practice is, in much of the crocodilian's range, enormous. A gap that reflects shortfalls in enforcement resources, in institutional capacity and the political will to prioritize wildlife protection against competing economic interests, and in the community support for conservation that makes self-enforcement possible. Addressing this gap requires not simply strengthening laws, but building the institutional capacity and community engagement that makes enforcement feasible and legitimate. The relationship between crocodilian conservation and broader governance quality in range countries is a correlation that conservation researchers have increasingly documented, but that conservation organizations have been reluctant to address directly because it requires engagement with political conditions that lie beyond the traditional scope of wildlife conservation. Countries with stronger rule of law, lower corruption, higher government effectiveness, and more accountable institutions tend to have better conservation outcomes for crocodilians and for wildlife more broadly, not because governance quality directly determines crocodilian abundance, but because governance quality determines the effectiveness of the regulatory frameworks and enforcement mechanisms that conservation depends on. This correlation has been documented across multiple taxa and multiple regions, and it suggests that investments in governance reform, transparency, accountability, institutional capacity building may be among the most cost-effective contributions to long-term conservation outcomes, even though they are not conventionally classified as conservation investments. The political economy of conservation funding, where money for wildlife conservation comes from, who controls it, and how it is allocated across competing conservation priorities, is a dimension of the conservation landscape that shapes outcomes in ways that are rarely discussed openly, but that are pervasively influential. The global conservation funding system is dominated by a relatively small number of large foundations, bilateral aid agencies, and international non-governmental organizations based primarily in North America and Europe, whose funding priorities and decision-making processes are inevitably shaped by the interests, values, distinct data, and political contexts of their home societies. This creates a structural asymmetry in which the priorities of wealthy country conservation donors do not always align with the priorities of the range country communities and institutions whose cooperation is essential for conservation success. Crocodilians illustrate this asymmetry clearly. In wealthy country conservation communities, the charismatic megafauna that attract the most funding, lions, elephants, tigers, great apes, are not crocodilians. Crocodilians are perceived, often accurately, as animals that generate fear rather than affection in general audiences, that lack the infant-like facial features that drive mammalian charisma and that do not easily support the emotional narratives of individual animal stories that drive conservation fundraising. The result is that crocodilian conservation is systematically underfunded relative to the ecological importance of the order and the conservation needs of its most endangered members. The gharial, one of the most critically endangered large vertebrates on Earth, receives a fraction of the conservation funding directed toward charismatic mammals of comparable conservation concern, and this funding gap has direct consequences for the speed and scale of conservation response. Addressing this funding asymmetry requires both better communication of the scientific and ecological case for crocodilian conservation, making the argument more effectively that these animals matter for reasons that should compel funding even in the absence of conventional charisma, and the development of alternative funding mechanisms that are less dependent on the emotional preferences of wealthy country donors. These alternative mechanisms include the payment for ecosystem services frameworks described earlier, the commercial sustainable use revenues from CITES-approved trade programs, the tourism revenues generated by crocodilian wildlife watching programs, and the potential biomedical research value that crocodilian physiology represents. None of these alternatives is sufficient on its own, and none is without its own complications and controversies. But together, they represent a funding base that is more diverse, more locally grounded, and more directly tied to the value that crocodilians provide to the ecosystems and communities in their range. a funding base that is therefore more sustainable in the long run than dependence on the charitable preferences of distant donors. The specific geography of crocodilian distribution in relation to human population centers and economic activity creates a spatial pattern of conservation opportunity and challenge that varies dramatically. Across the order's range, in the remote, sparsely populated regions of the Amazon basin, the Congo drainage, and the river systems of central New Guinea, crocodilian populations face relatively low, direct human pressure, but are threatened by the slow advance of agricultural and extractive frontiers that are progressively converting wilderness to managed landscape. Conservation in these areas is primarily a matter of maintaining the wilderness character of large areas against the economic pressures of frontier development, a challenge that requires engagement with national and regional economic planning processes and with the international trade systems that drive the commodity production motivating that development. In the heavily populated river systems of South and Southeast Asia, the Mekong, the Irrawaddy, the Salween, the rivers of the Indian subcontinent, crocodilian populations occupy landscapes where almost every square kilometer has been modified by centuries of intensive human land use, and where the few surviving crocodilian populations are restricted to the most inaccessible remnants of suitable habitat. Conservation in these landscapes requires not simply protecting what remains, but actively restoring habitat conditions. River flow regimes, water quality, bankside vegetation that have been degraded by agricultural and industrial development. This restoration challenge is technically complex, politically demanding, and expensive, and it requires partnerships with water management authorities, agricultural sector regulators, and industrial polluters whose cooperation is difficult to secure without the leverage of regulatory frameworks and economic incentives that conservation organizations alone cannot provide. In the urban and peri-urban landscapes where crocodilians increasingly encounter human populations, the waterways of northern Australian cities, the canals and drainage systems of Florida's expanding metropolitan areas, the rivers flowing through the expanding cities of Southeast Asia, the management challenge is one of physical and behavioral modification of an urban wildlife population whose presence is both ecologically significant and practically problematic. Urban crocodilians learn, sometimes rapidly, to exploit the food resources, garbage, domestic animals, fish from urban waterways associated with human settlements, and this learned behavior increases the frequency and proximity of human encounters in ways that elevate conflict risk. Managing urban crocodilian populations requires a combination of physical infrastructure, barriers at high-risk access points, clear signage about risk areas, and behavioral management of both the crocodilians and the human communities whose water-related activities bring them into the crocodile's operational range. The communication of crocodile risk to urban and peri-urban communities is an area where public health communication principles are directly relevant and where conservation organizations and wildlife management agencies have not always applied those principles as effectively as the situation demands. Risk communication research has established clearly that effective messages about environmental risks must be credible, specific, actionable, and culturally appropriate. That generic warnings about dangerous animals are far less effective than specific information about the times, places, and behaviors that create elevated risk, combined with concrete guidance about what individuals can do to reduce that risk. Applied to crocodile risk communication in urban settings, this means replacing the generic warning sign with specific information about the hours of greatest crocodile activity, the specific waterway sections where crocodiles have been recently observed, and the specific behaviors, swimming, fishing from low banks at dusk, allowing pets to access waterways that create the greatest risk. The engagement of media in crocodile risk communication creates dynamics that are both useful and problematic. Media coverage of crocodile attacks drives public awareness of the risk, which is useful for motivating protective behavior, but it also drives fear responses that can generate pressure for population reduction measures, culling, trapping, relocation, that are both ecologically harmful and practically ineffective at reducing attack risk in the long term. The management of media narratives around crocodile attacks requires wildlife management agencies to engage proactively with journalists and media organizations, providing scientific context for attack events, explaining the ecological significance of crocodilian populations, and offering alternative framings of the human-crocodile relationship that are accurate, nuanced, and less likely to generate the panic-driven demands for population suppression that simplistic predator attack narratives tend to produce. The role of zoos and aquaria in crocodilian conservation has evolved significantly over the past 3 decades, from institutions that primarily displayed crocodilians as spectacle, the prehistoric monster behind the glass, to institutions that are active participants in research, captive breeding, and conservation program support for endangered species. The Species Survival Plan programs, coordinated by the Association of Zoos and Aquariums in North America, and equivalent programs in European and Australian zoo networks, manage captive populations of endangered crocodilian species, the gharial, the Chinese alligator, the Philippine crocodile, the Cuban crocodile, the Siamese crocodile, the Orinoco crocodile, as genetic reserves whose long-term purpose is to maintain the genetic diversity and demographic viability of captive populations that can contribute individuals to wild population reintroduction programs. The management of these captive genetic reserves requires sophisticated pedigree analysis and breeding program coordination to avoid inbreeding while maintaining sufficient captive population size to buffer against demographic accidents. It requires veterinary expertise specific to crocodilian medicine, an emerging specialization that is producing protocols for anesthesia, nutrition, disease treatment, and reproductive management of captive crocodilians that improve both captive welfare and the viability of animals destined for reintroduction. And it requires sustained coordination between zoo programs and in-country conservation organizations to ensure that the captive genetic reserve is aligned with the needs of the Wild Population Recovery Program. It is designed to support a coordination that requires long-term institutional relationships, shared data systems, and mutual understanding of each partner's constraints and priorities. The educational function of zoo crocodilian exhibits is significant in ways that extend well beyond the simple entertainment value of seeing a large predator at close range. Well-designed exhibits that communicate accurate information about crocodilian biology, ecology, and conservation status can shift visitor attitudes from fear and disgust toward understanding and concern, a shift that, replicated across the millions of zoo visitors who encounter crocodilians each year in institutions around the world, contributes meaningfully to the broader public constituency for conservation. Research on visitor responses to zoo crocodilian exhibits has documented that exhibits that provide behavioral context, allowing visitors to observe natural behaviors like thermoregulation, social interaction, and feeding rather than simply viewing static animals, produce more positive attitude changes and more lasting knowledge retention than exhibits where animals are presented without behavioral context. This finding has influenced exhibit design in progressive zoos and has contributed to the movement toward naturalistic, behaviorally enriching captive environments that serve both animal welfare and conservation education goals. The intersection of crocodilian conservation with international development programs, the large-scale investments in infrastructure, agriculture, and economic development funded by multilateral development banks, bilateral aid agencies, and private investment that are transforming the economic landscapes of crocodilian range countries represents both one of the most significant threats to crocodilian habitat and one of the most important opportunities for mainstreaming conservation into deep development. Infrastructure investment, roads, dams, irrigation systems, ports creates the physical modifications of river systems and wetlands that are among the most consequential drivers of crocodilian habitat loss. Agricultural expansion, funded by international investment in commodity crop production, converts wetland habitats to farmland in ways that eliminate both the aquatic habitats that crocodilians depend on and the terrestrial buffer zones around those habitats that reduce human-wildlife conflict. But the same development institutions that fund these habitat-modifying investments also have environmental and social safeguard policies that, when adequately implemented, and require assessment of the impacts on wildlife and ecosystems and the adoption of mitigation measures to reduce those impacts. The quality of environmental impact assessment for development projects in crocodilian range countries varies enormously, from genuinely comprehensive, science-based assessments that identify significant crocodilian habitat impacts and recommend effective mitigation measures to superficial, checkbox exercises that identify no significant impacts regardless of the ecological reality. Improving the quality and enforceability of environmental impact assessment for development projects in crocodilian range countries is one of the most practically important contributions that conservation scientists can make to the long-term protection of crocodilian habitats more important in terms of habitat area affected than any number of protected area designations in areas that were not threatened by development in the first place. The contribution of crocodilians to nutrient cycling in freshwater and estuarine ecosystems is a dimension of their ecological function that is rarely discussed in conservation contexts, but that may be among the most ecologically significant things they do. Large predators that consume substantial quantities of prey and then defecate in water contribute to the movement of nutrients from terrestrial and aquatic food webs into the water column in forms that are available to primary producers, algae, aquatic plants, and to the invertebrate communities that form the base of aquatic food webs. Crocodilians, which feed extensively on fish, birds, and mammals and spend most of their time in or near water, are particularly effective at this nutrient transfer function. In river systems and wetlands where large crocodilian populations have been reduced or eliminated, the nutrient cycling contribution of these animals is lost, with potential consequences for the productivity and biodiversity of aquatic communities that are difficult to measure but real in their ecological significance. The role of crocodilians in seed dispersal, a function not typically associated with large aquatic predators, has been documented in tropical river systems where these animals consume fruit that falls into the water and subsequently deposit viable seeds at locations distant from the parent plant. While seed dispersal is not a primary ecological function of crocodilians in the way that it is for frugivorous mammals and birds, the incidental dispersal of seeds through the gut passage of animals that move extensively through river systems and between aquatic and terrestrial habitats adds a small but real component to the dispersal. Processes that determine plant community composition in riparian and wetland ecosystems. This function like others that crocodilians perform, is lost when populations decline, contributing to the suite of ecosystem services that are degraded when apex predators are removed from functional landscapes. The monitoring of crocodilian populations over time, the detection of trends in abundance, distribution, size structure, and reproductive success that indicate whether a population is stable, growing, or declining is a technical challenge that has been approached through a variety of methodological frameworks whose strengths and limitations are important to understand for interpreting the available data. Night spotlight surveys, in which a boat or shore-based observer moves through a defined water body at night, counting crocodilians detected by the eyeshine reflected from flashlights or spotlights, have been the standard population assessment method for crocodilians in aquatic habitats for decades. They provide a relatively standardized, repeatable index of population size that can be compared across years to detect trends, but they have important limitations. They detect only individuals that are in accessible, open water. During the survey period, they are influenced by weather conditions and observer experience in ways that are difficult to fully control. And they provide abundance indices rather than absolute population estimates. More sophisticated methods mark recapture programs in which individuals are physically captured, marked with unique identifiers such as scale clips or implanted tags, released and then detected again in subsequent surveys, provide absolute population estimates with quantifiable uncertainty. But they require the physical capture of animals, which is logistically demanding, potentially stressful to the animals, and in some cases impossible in habitats where capture techniques cannot be effectively deployed. The development of non-invasive individual identification methods using natural markings, photographic identification, and genetic identification from environmental samples is progressively reducing the need for physical capture while maintaining or improving the quality of population estimates. And the integration of these methods with drone-based survey platforms and AI-assisted image analysis is creating population monitoring systems of unprecedented efficiency and coverage. The long-term population monitoring programs that have been maintained for American alligators in Louisiana and Florida, for saltwater crocodiles in the Northern Territory of Australia, for Nile crocodiles in several African river systems, and for spectacled caimans in parts of South America, represent some of the most valuable datasets in crocodilian science. Datasets that span multiple decades that document population responses to changing management regimes and that provide the empirical foundation for understanding the demographic dynamics of these long-lived animals. Maintaining these long-term monitoring programs through changing funding environments, shifting institutional priorities, and the inevitable personnel changes that occur over timescales of decades is one of the most important and most underappreciated contributions to crocodilian conservation. The scientific value of a long-term dataset increases non-linearly with its duration. A 30-year time series is not simply 3 times more valuable than a 10-year time series. It is qualitatively different, capable of answering questions about population dynamics on timescales that shorter records cannot address, which means that the interruption of a long-term monitoring program represents a loss that is disproportionate to the immediate cost saving it achieves. The stories that individual crocodilians could tell, if we had the means to read their full life histories from birth to death, would be stories of extraordinary persistence and adaptability, of animals navigating landscapes of constant change and constant threat with a combination of innate behavioral programs and accumulated learned knowledge that proves, in most cases, adequate to the challenge. They would be stories of patience, The patience of an animal that can lie motionless for hours, waiting for an opportunity, that can go months without eating, without apparent distress, that can wait out a dry season in a shrinking pool, with the physiological stoicism of something that has been refined for exactly this contingency. They would be stories of power, the sudden, explosive, overwhelming power that 200 million years of predatory refinement can produce when the moment of action finally arrives. And they would be stories of continuity, of a lineage that has threaded its way through every catastrophe that the history of the planet has produced, maintaining its essential character while adapting at the margins to the specific conditions of each era, each ecosystem, each specific stretch of river or patch of wetland that has been, for some duration, home. These are stories worth telling, worth understanding, and worth the sustained human effort required to ensure that they continue to be written in the rivers and wetlands of a world that, despite everything, still contains one of its most ancient and most accomplished inhabitants. The crocodile has earned its 200 million years. The question of whether it earns the next 200 or the next 2,000 is a question that its biology alone cannot answer. It is a question that we must answer with the choices we make, with the priorities we set, and with the commitment we bring to the remarkable and irreplaceable living world we have inherited and that we now, for better or worse, hold in our hands.